Good evening. My name is Carroll and many of you know me. I'm the director of the UWA Institute of Agriculture. Before we get started, I wish to acknowledge that the university is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, belief, and knowledge. It's my pleasure to welcome you to the 2025 My Carroll Traveling Fellowship Award evening. I would like to especially acknowledge Mrs. Helen Carroll, her daughter Marie-Louise Carroll, granddaughters Coco Dibola, which she came last year as well, and the new member, Abby Dibola, who are here tonight. It's also wonderful to see here fellowship supporters, sponsors, colleagues, friends, and students. At least 2 students are here. My Carroll Traveling Fellowship at the UWA honors the late Dr. Mike Carroll, recognizing his dedication to agriculture. He was a student here, and later on, he became the Director General of then Department of Agriculture in Western Australia, especially new people who doesn't know that. He did a lot of work. His area of research was in plant nutrition, and then some of the things he has done is enormous, connecting Western Australia with the global partnership. And I had the privilege of working under his leadership when I completed my PhD from this university. So this fellowship was created by colleagues, friends, farmers, GRDC, and many others. Now it is in the 21st year, so I have been attending all those 21 years. The fellowship has supported 32 PhD students since the first recipient in 2003. We had a function in the Vice Chancellery. The late Professor Alan Robson was championing at that time. And those students have gone to different parts of the world, including in Western Australia, and achieving some significant achievements there in their career. So the fellowship has done some remarkable job. In fact, we are just in the process of establishing a similar fellowship, although I haven't got the details. I'm working with the Office of the Development and Alumni Relationship at UWA for Professor Alan Robson. So some of you will get that within the next couple of weeks, which I'm looking forward. So idea is that having the fellowship in terms of an annual Alan Robson Memorial Lecture in Agriculture. It is now my pleasure to welcome Mrs. Helen Carroll to introduce our first student presenters, and I acknowledge all of you. I didn't want to really name anyone, but certainly these are the most important people who cares about this fellowship and agriculture in general in Western Australia. So, Mrs. Carroll, to introduce our first student presenter, please.
Good evening, ladies and gentlemen. Please, would you let me know if you can't hear me at the back of the room? You can't. You can. Great. Okay. Now, I would like so much to thank you for coming. And of course, I always need to thank Sadiq, because he is the person who steers me in the right direction all the time. Now, it's hard to believe that it's been 21 years since the Michael Carroll Fellowship was established. And it's a thrill to hear that more than 30 postgraduate students from UWA have benefited from the fellowship since the first recipient was awarded. Now, for some basic information. It's hard to believe, as you can see, I have become increasingly frail over the last few years. Thanks, Marie-Louise. And while I continue, of course, to come to the fellowship for as long as I'm here, this will be my last year of getting up to speak. That's officially anyway. I will be handing that role over to, it says here, Michael's daughter. She's mine too. Our daughter. So it's Michael's daughter, Marie-Louise, and her brothers who can't attend tonight due to being overseas and interstate. But for now, I am here. And I would like to introduce the first recipient and speaker, Ruby Wiese. Ruby Wise. Okay, Ruby Wiese. Now, a little about Ruby. She grew up on a farm near Narrogin, Western Australia. She completed an undergraduate degree in agricultural and engineering sciences at UWA and has been here ever since. After completing her honours, Ruby was lucky enough to be given the opportunity by her supervisors to work on an industry-funded project aiming to develop a new method for harvesting subterranean clover seed. Now, the project spans both agriculture and engineering, and under the supervision of pasture scientists, Professor Megan Ryan. Is Megan here by the chance? Because I used to teach her Italian. I was going to check her out, okay. So the project spans both agriculture and engineering, and under the supervision of pastoral sciences, as I said, scientist Professor Megan Ryan and acting Professor Phil Nichols, as well as engineers, acting Professor Andrew Guzzomi and Dr. Mesley Wast. Ruby has been pursuing a PhD focused on this topic. Now, in October 24, Ruby attended the International Plant Phenotyping Symposium in Lincoln, Nebraska, in the USA, funded by the Mike Carroll Travelling Fellowship. That is very important for me, and I wish my daughter to go ahead as she always does, and she achieves a lot. And I thank her for putting up with lots of things, but I know now that she has two little ones to assist. Thank you, ladies and gentlemen. It's so lovely to see familiar faces. Okay, thank you. Yes. Oh, I love this. Okay.
Helen. It is on. Do I just need to speak louder?
Yeah, maybe a little bit. Yes, maybe a little bit. Yeah.
Okay. Thank you all so much for being here to listen and letting me talk about this really amazing trip I got to go on, thanks to the Mike Carroll Travelling Fellowship. So I'd like to start off today by introducing my PhD topic. In short, I'm part of a larger project that aims to develop a new way to harvest subterranean clover seed. So subterranean clover, or subclover, is a pasture legume, and it's a really important and popular one, in part due to its trait of shallowly burying its seeds underground, hence the name subterranean clover. It's not working? Okay. However, this seed burial trait means that subclover seed can't be harvested using conventional combine harvesters. And instead, the industry relies on what are essentially huge vacuum cleaners to suck up the seed after multiple passes of preparatory tillage. This current practice works well, but it is inefficient, it relies on outdated machinery, and it causes quite significant soil degradation and erosion. So we've been looking at a swathing technique as an alternative, which involves cutting the plants before they have fully dried out in the field, and in this case, cutting them below the ground, forming them into windrows to finish drying, and then coming back and collecting them with a combine harvester. And we've taken inspiration for this sort of subterranean swathing method from the peanut industry. So during my PhD, I'm aiming to examine the feasibility of a swathing method for harvesting subclover seed and also to help guide its implementation. In particular, because swathing occurs much earlier than conventional subclover seed harvesting, I plan to examine how plant and seed characteristics change together in order to determine when the optimal time for swathing is. So during my PhD, I also want to learn some skills related to high throughput phenotyping and other imaging techniques, because this is becoming increasingly widespread and important in agriculture, and because I think it's a really interesting and exciting space. So to help me start to facilitate this, get some ideas for how I can incorporate this learning into my PhD and meet potential collaborators, I decided that I would like to attend the International Plant Phenotyping Symposium, which was held in Lincoln, Nebraska in the USA. And thanks to the Mike Carroll Travelling Fellowship, I was actually able to do this. Then, because it's such a long flight over to the USA, I wanted to make the most of this opportunity. So I also organised to meet 2 pastures researchers over there so that I can expand my network and learn about their research. And this also gave me an opportunity to get an understanding of agriculture in these 2 different places, which was something I find really interesting. So my first stop in the US was the University of New Hampshire, where I met Professor Richard Smith, whose research focuses on both agriculture and ecosystem ecology. Around 70% of the land in New Hampshire is forest, and I was lucky enough to be there at the start of fall, which meant that the leaves were just starting to change colour, and it was unlike anything I'd ever seen before. New Hampshire has a summer growing season, in contrast to WA. Many of the farms are small, and around half of their land is often forest. The farms are dominated by horticultural production, and a lot of the produce is just sold locally, for example, at farmers markets. So Richard does quite a lot of research on cover crops. These are crops which farmers plant when there isn't another crop growing, and its purpose is purely to cover the ground, preventing erosion and outcompeting weeds, with its biomass eventually returning to the ground rather than being harvested. This was not a concept I was familiar with at all, and it felt somewhat crazy to me coming from a wheat belt farm. And there was also a really diverse range of species that they were using as cover crops, which was amazing to see them all together and see that biodiversity. Then Richard also showed me another project he was working on involving a type of agroforestry called silvopastures, where both pasture and trees grow on the same piece of land. Currently, in other locations, silvopastures are generally established by planting trees into an existing pasture. However, because New Hampshire is already so forested, Richard was examining the concept of partially clearing some forested land and then establishing a pasture amongst these existing trees. So after New Hampshire, I headed over to Lincoln, Nebraska to attend the International Plant Phenotyping Symposium. The symposium was really valuable. It was awesome to see people from all over the world discussing their research, projects, and infrastructure, all relating to plant phenotyping. There's really so much going on in this space, and it was amazing to be a part of it. I also saw lots of ideas which I thought I might be able to apply to my project, so I'm looking forward to diving into these a bit deeper. One of the surprising things I discovered during the symposium was that the Australian Plant Phenomics Network is sort of leading the way internationally, and they have some pretty cool equipment, which was really good to know. My final stop was Oregon State University, where I met Associate Professor Serkan Ates, whose research focuses on pasture management. Corvallis, Oregon has a Mediterranean climate similar to WA. The area does quite a lot of pasture seed production, which is relevant to my PhD, but they also have a huge diversity of other agricultural industries. So I got to visit a small-scale seed production company owned by a farmer, where I saw lots of pasture grasses, a bit of clover, and even a paddock of chicory, a pasture species I'd never heard of before. I also visited a larger seed production company that does quite a lot of their own research, and an even larger company called DLF Seeds. I learned that this region used to grow quite a bit of subclover seed, but without an established and sustainable method for harvesting it, this has mostly stopped, and as a result, livestock farmers in the area don't tend to plant it. So it was interesting to think that once my project team develops a more widely accessible method for harvesting subclover seed, there's an opportunity for it to move back into some of these international spaces. Altogether, this trip was incredible. I really loved seeing new and different ways of doing things, and I feel like I learnt so much. I want to stress just how grateful I am to Richard and Serkan for giving me so much of their time to show me their states and lots of cool research. The conference was incredibly valuable for my future endeavours in plant phenomics, and I'm hugely grateful for the opportunity to have attended it. So most of all, I would like to thank the Mike Carroll Travelling Fellowship for facilitating this entire incredible experience for me. And thank you all for listening.
Okay, so thank you for that interesting and exciting talk. When we gave the fellowship, we didn't know all these things. I would have taken that fellowship and myself gone there. Look, any questions to Ruby? Yeah, Phil Werko, please.
Yeah. I did, yeah. There was a lot of presenters, I guess, from the States and also from Europe. And I mean, Europe tends to have quite a lot of cutting edge and cool technologies and labs. So there was a couple of, labs or even companies, I guess, in Europe that looked really interesting. I'd kind of gone over there to find, somewhere that had cutting edge technology that I could use, and ended up finding that Australia had some of the best stuff. But it looks like there's some interesting places in Europe that I'd love to visit, but I also loved the States. So yeah, I'm kind of just exploring my options, but it's really exciting.
Any other questions?
No, they certainly had some, I think, white and red clover. So they had other clovers that could be harvested, and they had like quite a diverse range of pasture species, but yeah, lots of grasses as well. But it seems to mostly be replaced by other legumes that are easier to harvest, such as other clovers.
All right. If there are no other questions, thank you very much for that interesting talk, and wish you all the best for the remainder of your PhD program. Now, I would like to invite Marie-Louise Carroll to introduce the second speaker. Thank you.
Thank you, Sadiq, and good evening, everyone. And thank you all for coming. So it's my pleasure to introduce our next speaker, whose name I will try and pronounce correctly, Agyeya Pratap. Agyeya is a biotechnology postgraduate from India with experience in studying abiotic and biotic stresses in multiple crops, including wheat, rice, tomato, and mustard. In 2020, he was awarded a fully funded PhD at UWA to pursue his passion for crop improvement. but due to COVID-19 travel restrictions, he commenced his PhD offshore in collaboration with the Indian Agricultural Research Institute. Under the supervision of Professor Sadiq, his research focuses on wheat heat tolerance, examining phenology, physiology, yield, and key agronomic traits in contrasting wheat genotypes, which I'm guessing, unless you're from an agricultural background, that probably needs to be unpacked, which I'm sure will happen. His academic journey has been enriched by prestigious opportunities, including the Mike Carroll Travelling Fellowship, which supported his participation at the UWA Plant Energy Management, Molecular Mechanisms and Signalling 2024 in Sweden and Tropentag 2024 in Austria. Additionally, he collaborated with Dr. Jiang Feng Mao, hopefully I've said that correctly, at the UWA Plant Science Centre in Sweden. Please welcome Agyeya.
Thank you for the warm welcome, and hello everybody. Thank you for coming here and taking out time from your busy schedule. So my name is Agyeya Pratap. I'm a PhD student at University of Western Australia under supervision of Professor Kadambod Siddiq and Associate Professor Nicholas Taylor. So in collaboration with Indian Agriculture Research Institute, I did some part of my research in India due to COVID-19 under supervision of Professor Madan Pal and Viswanathan Chinnuswamy. With the help of my Carroll Traveling Fellowship, I was able to attend these two conferences, wonderful opportunities in Europe, one in Sweden and the other one in Austria. My PhD in University of Western Australia focuses on unraveling heat tolerance in bread wheat with insights from flag leaves and spike tissues, which are major components of drivers of yield in wheat, which basically deals with adaptation of wheat to climate change, mitigating yield losses due to increasing frequency of heat waves, and while finding protein biomarkers for heat tolerance, which will help in identifying heat tolerant wheat genotypes in the future breeding programs. Now, this study deals with a lot of technical aspects of proteomics, which might not be good for discussion over here. But then I specifically deal with studying changes in protein abundances in flag leaves or leaf tissue of wheat and spike tissue of wheat, and looking at quantitative changes between these protein members and seeing if there are any specific pathways that we can identify that might help us in identifying heat tolerant genotypes in future. Now, in Sweden, I attended the UWA Plant Energy Management Molecular Mechanisms and Signaling Conference. Such a long name, but yeah, it covers all of that. There were speakers from all around the world, including countries like Japan, Germany, Belgium, France, UK, the USA, and they all they are literally leaders in their research fields, especially in plant development, plant nutrition, plant flower development, even for tuber crops and many other crops, including agroforestry. So such a diverse group of speakers, where I gave an oral presentation and also gave a poster presentation during this conference. I got into discussions with many of these experts and got some great insights on how I can at least look or change my perspective on my research. I got great insights on how experimental design might have affected the results that I've found, and that also helped me question and also helped me gain more confidence in my future career for my future career in plant science. During this conference, I also attended various research facilities at the UWA Plant Science Center. They have state-of-the-art facilities which provide, for example, semi-automated plant phenotyping facility, GMO plant growth facility, and atomic force microscopy. The semi-automated plant phenotyping facility helps us accelerate uh, the measurements of plant traits, which might be of use to different uh, crop research programs. The GMO plant growth facility helps us identify or at least characterize gene functions in plants by creating mutants. The atomic force microscopy helps us study structures of plant cells at a very detailed scale. That is definitely helpful in characterizing specific gene functions also when we are creating mutants. After this conference, I collaborated with Dr. Jianfeng Mao, same at the UWA Plant Science Center, where the purpose of my lab visit was to design an in silico allele-specific gene expression analysis pipeline. Now, this pipeline will definitely help us pinpoint alleles with greater detail, which might have some role in heat stress tolerance in wheat. During this 1 week visit, we were able to create the project design, had extensive discussions on my study design, everything else. We even were able to find the tools that might be useful in developing that pipeline. And by day 5, we were actually able to start developing the pipeline. Although the visit was really short, but we decided that we will continue this collaboration, and at some point, we will be able to publish this work in the future. After that, I went to Austria to attend the Tropentag 2024. This conference deals with agroecology, rural development, and agronomy, and sustainable development. Now, they had talks related to agroforestry, a lot of workshops on capacity building, and at the same time, talks on abiotic stresses. In one of which sections, I presented my poster on heat tolerance in bred wheat. I got really good responses from many people, from including Professor Volkard Asch, who's a leading researcher in Germany on abiotic stress tolerance in crops. At the last day, on the last day of this conference, I got the opportunity to attend this wonderful workshop on biological nitrification inhibition of wheat, which deals with improving the nitrogen use efficiency in wheat. So this workshop was carefully designed by experts from across the globe who are dealing with at least some aspect of plant, environment, and genotype interactions. I also got to meet Dr. Wolfram Weckreath, who has a lab in Vienna, University of Vienna School of Biological Sciences. We had a very great discussion about my own work and his own work on studying different aspects of plant microbiome, soil microbiome, root interactions, and plant interactions, and how nutrition is affected by all three. So I would like to thank Mike Carroll Traveling Fellowship for providing me this great opportunity for traveling to Europe. This was my first time traveling to Europe, actually. So thank you so much for that.
Hi, does everyone have any questions for this gentleman whose name I hope I pronounced Agyeya? Does anyone have any questions for Agyeya?
There are various ways to do that. One way is to screen the current existing germplasm and see which genotypes express that gene in a higher composition. Meanwhile, at the same time, we have technology today that can easily transform any plant to express any of these genes at a higher concentration.
Is it a correlation, or if you put the gene in as you convert, it's expressed? I'm sorry, I'm still having trouble understanding how you get a positive effect.
So first, identification of these genes requires us to study the existing germplasm and see if some traits are associated with higher expression of some genes. Once we have done that and identified those genes, we can actually change the precursor sequence in the DNA of any gene, which can express the genes to a higher level compared to original plant that we would have that we had. So, and then we can study like what effects it has on the plant development, plant traits, and yield, and etc.
Okay, thank you very much.
Thank you.
Thank you, Marie-Louise, and also the students for their wonderful talk. And I'm sure that the intent of the fellowship was to attend some conferences, go to some lab, the time period is short, and use those information either for their thesis, because they haven't submitted the thesis. Agyeya is submitting on 30th of April. Ruby, when you are submitting? 6, so you've got quite a long way to go. So certainly I can see that I'm just going through his chapters and so on, so I can see some of those aspects are being dealt. So, certainly it has helped. And certainly the future of these students in terms of getting that international network, which Phil has mentioned, and the confidence build up, attending these conferences and things. When we were students, we had not such opportunities, to travel and so on. But that's good, and please give them, again, appreciation. And what we do when we call for the next, that is 2025 round, or whatever round we have been calling, we got a small committee, and that committee is chaired by Helen, and we also have Terry and Wright. Before that, we had Dave Chattel, so who is could not make it. So Terry is our chair of the Industry Advisory Board, whether he come online or sometimes he come in person. Myself, and assisted by Cora Castens, our executive assistant. She was there, and we do get half a dozen to 8 applications, and we go through critically and then make the decision. And this year's fellowship, I think, closes on 31st of May, and then we will go through that and do. So it has been a great initiative, and it is still continuing, which is really good, and no one can take that money from the university for other purpose. That is the beauty about these Beckers funds. Only thing is that someone has to follow behind and get this going, and we have been able to do that for the last 21 years. Well, we probably have had bit arguments and so on, but certainly I think we got the right topics and so on. So thank you very much again for attending this evening, and your continued support in coming and supporting this is greatly appreciated. I was just talking with the Vice Chancellor yesterday and saying that we need more support like this for students. The more we can give better experience for them in the university. Now with that, I would like to invite you to join in the foyer for nibbles and some more drinks. And we also have various functions. 28th May is our 19th postgraduate showcase, and Governor, His Excellency Governor of Western Australia, is coming for that. And there's a number of other events, including Industry Forum. We got the Hector and Andrew Stewart Memorial Lecture, Brian Carlin Memorial Lecture. So quite a lot of them are coming. Certainly keep an eye on the information coming from the Institute. I also want to thank Anna. Anna is our new recruit. You may remember Rosanna. Rosanna gone on a maternity leave. She had a lovely daughter a month ago, and Anna is now stepping in her shoes and doing a wonderful job. And we got here Diana, our business manager, So it'll be like a small team working to achieve all these things. So thank you all my colleagues, and Wallace Cowling, my associate director, is also here, and Phil Werko as well. Yeah, sorry. Thank you again. Let us have some drinks. The photos are okay? Yeah. How are you going to manage? Can we go in back?